7 states across this country have passed laws making it illegal to sell a specific food product to their citizens. Florida, Texas, Alabama, Mississippi, Nebraska, Montana, Indiana. 7 independent state governments looked at the available scientific evidence and said, not yet, not until we have answers to questions that nobody in the industry wants to ask. And yet the federal government cleared that same product for sale nationally in 2023, not based on 20-year longitudinal studies, not based on randomized controlled trials following human populations across decades, based on a document review process using safety data submitted by the companies with the largest financial stake in getting it approved quickly. That product is cultivated meat. And before you decide what you think about it, I want you to hear what the peer-reviewed science actually says, not the press releases, not the investor presentations, not the political commentary from either direction, the published research. The words of the researchers themselves. In the next 15 minutes, I am going to show you exactly what cultivated meat is at the cellular and genetic level, why the specific biological processes required to produce it at commercial scale connect directly to pathways that matter enormously in the aging human body, and what you can eat instead to actively support the regenerative capacity you already have. This is not a political video. I do not care which party you support. What I care about is what the science says, and right now the science raises questions that the mainstream food conversation is not answering honestly. Before we go further, drop a comment right now with just your age, nothing else, just your age, because what I am about to share applies very differently depending on whether you are 52 or 72. The biological reasons for that difference are at the center of everything we are going to cover today. And if you have noticed increased fatigue, slower recovery after physical activity, or more frequent infections over the past 2 to 3 years, add the word slower to your comment. That is information I want to see because it connects directly to what I am about to explain about your stem cells. Picture the way this technology was introduced to the public. Clean visuals, the word sustainable repeated in every headline, promises of protein without slaughter, meat without land use, a solution to some of the most genuinely difficult problems in global food production. The average person reading those stories had no way to evaluate what was happening at the biological level inside those production facilities, because the industry did not put that information in the press releases. It ended up in peer-reviewed journals that most people never read, and the gap between what the research actually shows and what is being communicated to the consumers who will be eating this product daily across years and decades is, in my view, one of the most serious transparency failures in modern food policy. The research on the genetic processes involved in producing cultivated meat at commercial scale, specifically what happens to the cells that become the food, should have generated serious regulatory questions before commercial authorization was granted. Instead, it was cleared 7 state governments reviewed the same evidence and reached a different conclusion. That disagreement is not political theater. It is a substantive regulatory dispute grounded in published science that deserves honest examination. Now, if you are someone who wants to understand the why, not just a conclusion, then what I am about to explain will give you a completely different framework for thinking about this issue. Every healthy cell in your body operates within a biological constraint called the Hayflick limit. Think of it as a built-in division counter. Each cell can copy itself only a limited number of times before it stops dividing. This ceiling exists for a reason that is fundamentally protective. Cells that divide indefinitely without a stopping point are almost by definition pathological. The clinical term for what happens when a population of cells loses that constraint and begins replicating without stopping is malignant transformation. In plain language, that is how cancer begins. To produce cultivated meat at commercial scale, manufacturers need cells that ignore this limit. They need cells that will keep dividing day after day, week after week, filling industrial bioreactors continuously. That requires cells to be modified either through deliberate genetic engineering or through extended laboratory culture to become what scientists call immortalized cell lines. Lines of cells that do not stop dividing. Now layer on to that a second biological change that comes with normal aging. Your own stem cells, the ones your body uses to repair tissue, renew organs, and maintain biological function, carry what you could describe as an accumulated record of every division they have performed across your lifetime. Every round of cell division introduces a small probability of a replication error that is not corrected before becoming permanent. Your DNA repair machinery catches the vast majority of these, but some accumulate over time. The older you are, the higher your baseline mutational load in your stem cell populations, and the less efficiently your DNA damage response systems operate. Think of it like a quality control inspector who has been working 12-hour shifts for 30 years, still doing the job, but catching fewer errors per shift than they did at 30. The efficiency of your primary tumor suppressor system decreases measurably with age. This is one of the primary biological reasons why cancer incidence rises so sharply in the 6th, 7th, and 8th decades of life. These are not small differences between a 45-year-old and a 65-year-old. These are fundamental changes that demand a completely different framework for evaluating any novel food technology. And yet the guidelines used to authorize this product were not written with that framework in mind. Let me take you into the specific published science. A 2024 paper published in Frontiers in Nutrition examined the challenge of producing cultivated meat at industrial scale. The researchers stated directly and without ambiguity that to achieve the continuous cell division required for commercial production, the natural biological brakes on cell growth need to be overcome. The approaches described include altering genes that regulate the cell division cycle, specifically genes like TP53 and PTEN. I need you to understand what those genes actually do, because they are not obscure regulatory details. They are central to everything that follows. TP53 is the most studied tumor suppressor gene in all of cancer biology. It is sometimes described as the guardian of the genome, and that description is accurate when your DNA sustains damage from radiation, from environmental toxins, or from errors that accumulate during routine cell division. TP53 is the molecular mechanism that detects that damage. Once it detects it, it does one of two things. It pauses the cell division cycle to allow repair machinery to correct the error, or it triggers programmed cell death to prevent a damaged cell from replicating and passing its errors forward. TP53 mutations are found in more than 50% of all human cancers across all tissue types. PTEN is the second gene named in that paper. It is another tumor suppressor that regulates cell proliferation and survival. When PTEN function is reduced, cells receive a persistent pro-growth signal that drives them toward uncontrolled division. Here is the phrase from the published research that should command your attention. The paper states that this genetic alteration can be achieved spontaneously by prolonged culturing. You do not have to deliberately engineer these mutations in. Leave cells in culture long enough under the conditions required for industrial production, and they start accumulating these mutations on their own. The production environment itself creates selective pressure that favors cells that have already partially lost the genetic constraints on unlimited division. A separate 2023 paper in the same journal found that normal cells from non-primate animals, the type most commonly used in cultivated meat production, develop what the researchers describe as a tumorigenic profile after only 2 passages in a laboratory environment. 2 rounds of cell division in culture, these were cells that began as normal, healthy, functional tissue. Within 2 generations of laboratory culturing, they were already showing genetic characteristics consistent with tumor cell behavior, including evidence of metastatic potential. That recommendation has not been adopted as a regulatory requirement by any major food authority. The second mechanism deserves equally careful attention, and this one connects to something you may already know from longevity research. To keep cells viable and continuously proliferating in bioreactors across commercial production timelines, cultivated meat manufacturing requires continuous delivery of growth factors, specifically insulin-like growth factor and epidermal growth factor. These are molecular signals that tell cells to grow, divide, and survive. In normal human physiology, these signals are tightly regulated. Your body produces them in response to specific physiological triggers, exercise, tissue repair, growth states, and then reduces them when the need has resolved. That pulsatile, context-dependent pattern of signaling is physiologically important. It is not the same as having these growth factors present continuously at the concentrations required to sustain indefinite cell proliferation. The question that has not been answered is this: does residual growth factor activity persist in the final cultivated product? And does regular ingestion of cells that have been continuously saturated with pro-proliferative signals have any measurable downstream effect on the growth factor environment of the person consuming them over months and years? The reason this matters most for people over 50 is that the relationship between circulating IGF-1 levels and cancer risk in older adults is one of the most thoroughly documented associations in cancer epidemiology. The data is consistent across multiple large cohort studies and has been the subject of major meta-analyses published in high-impact journals, including The Lancet Oncology. Elevated IGF-1 is consistently associated with significantly increased risk of prostate cancer, breast cancer, and colorectal cancer, with the strength of that association increasing in people over 50. The mechanism is well characterized, IGF. One activates an intracellular signaling pathway that promotes cell survival and actively suppresses the programmed death processes that normally eliminate cells showing early premalignant changes. In a body where tumor suppressor function is already reduced by normal aging, persistent activation of this pro-survival pathway in the wrong cellular context is precisely the mechanistic sequence through which malignant transformation progresses. I am not claiming cultivated meat causes cancer. The published data does not support that statement, and I will not make claims the data does not support. What I am stating, based on what is actually in the literature, is that the specific biological processes required to produce this food involve the genetic and molecular pathways that 6 decades of cancer research have identified as centrally relevant to malignant transformation in aging human biology. And there is no published long-term safety data on human populations consuming this product across years or decades of regular exposure. The third concern entered the public record through the Texas legislative process and connects to one of the most rapidly growing areas of environmental health research, microplastics. Industrial bioreactors are manufactured from plastic. The cells that become cultivated meat are grown in direct, sustained, continuous contact with plastic surfaces, plastic-lined vessel walls, and in many production systems, plastic-based scaffolding materials that provide the structural framework on which muscle cells organize into tissue. Members of the Texas legislature specifically cited as part of the documented rationale supporting their prohibition that the production process involves direct interaction between cellular matter and microplastic materials with potential for disruption of cellular membrane integrity. Over the past 3 years, the peer-reviewed literature on microplastic biology has expanded dramatically. Microplastic particles have been detected in human blood samples, pulmonary tissue biopsies, arterial plaque specimens, and human placental tissue. The interaction between microplastic particles and mammalian cell membrane function is an active and rapidly expanding area of investigation, and the characterization of microplastic incorporation in the final cultivated meat product has not been published in the peer-reviewed literature. There is also a fourth concern that receives less attention but is in some ways more immediately relevant to how you feel and function day to day. Real animal food, specifically grass-fed, pasture-raised meat, is not simply protein in a structural matrix. It is a biologically complex substrate. It contains active enzymes that support digestion. It contains connective tissue rich in glycine, proline, and hydroxyproline, amino acids that act as signaling compounds for the stem cells involved in maintaining the integrity of joints, bones, tendons, and the gut lining. The fat in grass-fed beef carries conjugated linoleic acid at concentrations 2 to 5 times higher than grain-fed beef, with documented activity on tumor suppressor pathway activation in peer-reviewed research. Cultivated meat grown in a sterile bioreactor is biologically impoverished by comparison. It is muscle fiber architecture without an ecosystem. It is protein without the biological intelligence that real food has carried into the human diet across thousands of years. For someone over 50, where gut microbiome diversity has already declined, where digestive enzyme production is already decreased, substituting a food your biology has a 10,000-year relationship with for something that has existed for under a decade, without long-term safety characterization, is not a nutritionally neutral act. Here is where I want to stop and say something directly to you. If you have been trying to make careful, health-conscious dietary decisions, if you have been reading labels cutting processed food, paying attention to what you eat, you are the person for whom this evidentiary gap matters most. Not someone who eats fast food 3 times a day and gives no thought to any of it. You. Someone who is specifically trying to make better choices. You now understand something that most people eating this product will never be told. You understand what the published research actually says about the genetic and molecular processes involved in producing it. You understand why your age is not a minor variable in this conversation. And you understand that 7 state governments reviewed the same evidence and reached a different conclusion than the federal agency that cleared it for national sale. The federal government cleared it. 7 states prohibited it. I will leave you to draw your own conclusions about whose interest each of those decisions was primarily protecting. What no regulatory body could take from you is what you choose to put on your plate every day. Let me give you the practical answer, because the theoretical concern only matters if you have a concrete alternative. Here is what the published science supports for people over 50 who want to actively protect their own regenerative capacity. Grass-fed beef 3 to 4 times weekly, 85 to 140 grams per serving. Do not trim the fat. The conjugated linoleic acid and vitamin K2 and MK4 form that make grass-fed beef nutritionally distinct from grain-fed. Both reside in the fat fraction. Bone broth, made from collagen-rich bones, knuckle joints, femur with marrow, chicken feet, releases glycine, proline, and hydroxyproline in concentrations difficult to replicate through other dietary means. These amino acids are direct signaling precursors for stem cell maintenance pathways involved in preserving joint cartilage, bone density, and gut barrier integrity. 1 to 2 cups daily before your first meal, minimum 12 hours simmering for poultry bones, 24 hours for beef bones, beef liver, once weekly. The nutritional density of beef liver is without parallel in the conventional food supply. It is the most concentrated dietary source of CoQ10 and the most bioavailable dietary source of vitamin B12. B12 is a cofactor required for DNA synthesis in actively dividing cells, essential for the bone marrow stem cells that produce every red blood cell, white blood cell, and platelet in your body. B12 deficiency in people over 50 is found in 30 to 40% of this age group in epidemiological surveys. 100 grams once weekly provides over 400% of the recommended daily intake. Small fatty fish, sardines, mackerel, anchovies, herring, 3 to 4 times weekly, with soft edible bones intact where possible. EPA and DHA reduce pro-inflammatory signaling in bone marrow microenvironments where stem cells are housed. Vitamin D3 from fish fat directs stem cell differentiation toward bone-forming activity rather than fat accumulation, a balance that shifts unfavorably in most people after 50. Pasture-raised whole eggs 2 to 3 daily. Choline is required for DNA methylation and cell membrane synthesis. Chronic dietary choline insufficiency in people over 50 is extremely common, and it is associated with progressive disruption of the epigenetic regulation patterns that allow stem cells to maintain their identity over time. Fermented foods, raw sauerkraut, kimchi, plain whole milk yogurt, 2 to 3 tablespoons with meals. The organisms in properly prepared fermented foods produce butyrate through gut fermentation modulators in the current literature. It helps maintain the epigenetic stability in intestinal cells that keeps them behaving normally across decades of division. Wild blueberries, half a cup 3 to 4 times weekly, fresh or frozen. The compounds in wild varieties at higher concentrations than cultivated blueberries activate mechanisms that regulate stem cell quiescence, the precisely maintained resting state that preserves regenerative capacity across decades. Start with 3 of these, not all 7 simultaneously. Maintain those 3 consistently for 30 days. Then layer in the rest. Your gut microbiome, mitochondrial function, and inflammatory markers all respond to sustained dietary change, but on biological timescales of weeks to months, not days. One clinical note: if you are on warfarin, significant increases in dietary vitamin K from organ meats or fermented vegetables can affect your INR. If you have chronic kidney disease, increased protein intake needs calibration with your specialist. And if you are under active cancer treatment, none of these changes should be implemented without direct involvement of your oncologist. We have seen this pattern before. Trans fats were introduced as a healthier alternative to animal fats and remained in the food supply for more than 50 years. While the cardiovascular evidence accumulated, high fructose corn syrup was adopted at industrial scale before the long-term data on fructose metabolism was collected. Industrial seed oils displaced animal fats based on a hypothesis about saturated fat driven more by institutional momentum than by the quality of the underlying evidence. In each case, economic momentum moved faster than the safety science, and very large populations experienced the health consequences over decades before a reckoning was forced. Cultivated meat may ultimately prove entirely safe for regular long-term consumption. I genuinely do not know, and neither does any scientist currently working in this field. The long-term safety data required to answer that question definitively does not yet exist. What I do know is this: the biological processes required to produce cultivated meat at commercial scale involve the same genetic and molecular pathways, TP53 alteration, growth factor dysregulation, immortalization of cell lines through suppressor gene modification that 6 decades of cancer biology have identified as centrally relevant to malignant transformation in aging human tissues. 7 state governments examined the available evidence and determined the safety case had not been adequately established. The federal government decided otherwise. Those are the facts, clearly and completely stated. Keep asking the questions that press releases are not designed to answer. Drop a comment right now saying, "I informed that this video gave you something real." It tells me this research is reaching the people who need it most. Think of one specific person you know who is making careful dietary decisions and has never heard any of this. Send them this video right now, not later. Subscribe to this channel. My mission is to give you honest, research-grounded health information that most people only encounter after something has already gone wrong. You deserve to know it before that happens. In my next video, I am going to cover the specific blood test markers that give you the most accurate picture of your biological age, not your chronological age, and what you can do in the next 90 days to meaningfully shift those numbers. You will not want to miss it. This video is for educational and informational purposes only. It is not intended as medical advice. Dietary decisions must be individualized based on your complete medical history and current health status. Never adjust or discontinue any medication without first consulting your healthcare provider.